Welcome back everybody to Bobo's Biology channel, where we help you become an academic weapon just before your exam. On today's episode, we'll discuss the regulation of glycolysis. In this episode, we'll give you an introduction on the types of regulations present in glycolysis, the regulation of hexokinase or glucokinase, the regulation of Phosphofructokinase 1, the regulation of Pyruvate kinase, and at the end, to test your knowledge, we have included a new segment called Bobos Brain Busters. Feel free to skip to these timestamps in order to review the topic you need. If you're already familiar with the general types of regulation, please skip to the regulation of hexokinase or glucokinase. Let us start by telling a little story about a true experience of Bobo the Curious Mouse. One day, while exploring the forest, Bobo the Curious Mouse came across a river where his old school friend Barry the Beaver was contemplating. Barry the Beaver is a civil engineer specializing in dam construction. Barry the Beaver carefully places sticks and mud, letting just the right amount of water flow through. If the river was too fast, he adds more logs to slow it down, but if the water got too low, he removes some branches to let more water through. Watching this, Bobo the Curious Mouse had a eureka moment. Thanks, Barry, the civil engineer Beaver. You helped me figure out an analogy for glycolysis regulation. There are two main forms of regulation: allosteric regulation and hormonal regulation. Let's start with the allosteric regulations. Under normal circumstances, a substrate would bind to the active site to form the enzyme-substrate complex. When the complex dissociates, the product is released. The substrate is represented as S, and the product is represented as P. Allosteric regulation is where an inhibitor or activator binds to an enzyme at a site other than its active site, called allosteric site. This alters the shape of the active site and either enhances, in the case of an activator, or reduces, in the case of an inhibitor, the affinity of the enzyme for the substrate. Therefore, the activity of the enzyme is regulated. This regulation is fast and reversible, hence it can adapt quickly to cellular needs. The allosteric molecule can come from various points in the metabolic pathway. This means the allosteric regulator can act as a feedback inhibitor or feed-forward activator. In feedback inhibition, a product from later steps in the enzymatic pathway inhibits an earlier enzyme via the allosteric site, typically through allosteric inhibition or other inhibitory mechanisms. In the case of glycolysis, however, only feedback inhibition via allosteric regulation is relevant. This process prevents the overaccumulation of the final product, ensuring that valuable substrates are preserved for when they are truly needed. Remember the story of Bobo the Curious Mouse? When too much water was flowing, Barry, the civil engineer Beaver, would place sticks to inhibit the water flow. Feed-forward activation is where an earlier intermediate from the pathway stimulates a later enzyme to enhance efficiency and ensuring the pathway continues smoothly when needed. In the story of Bobo the Curious Mouse, when too little water was flowing, Barry, the civil engineer Beaver, would remove sticks to promote the water flow. The other form of regulation we will discuss is hormonal regulation. Insulin is released when blood glucose levels are high, and glucagon is released when blood glucose levels are low. We like to think of it as when glucose levels are low, then glucose is gone. Insulin works by causing dephosphorylation of the enzyme, which will lead to its inactivation or activation depending on the type of enzyme. Glucagon works by causing phosphorylation of the enzyme, which will lead to its inactivation or activation depending on the type of enzyme. This modification can activate or deactivate enzymes, providing long-term systemic control of metabolic processes. So, let's begin with the regulation of glycolysis. Glycolysis has 3 points of control: hexokinase or glucokinase, Phosphofructokinase 1, and Pyruvate kinase. The first point of control is hexokinase. Hexokinase converts glucose into glucose 6-phosphate. The phosphate tag traps it inside of the cell. Hexokinase is inhibited by glucose 6-phosphate, preventing glucose from being trapped in glycolysis when enough is already being used. Since the enzyme is being inhibited by the product of the enzyme, this form of inhibition is called feedback inhibition. Glucose 6-phosphate inhibits hexokinase by binding to an allosteric site and therefore is an allosteric inhibitor. An interesting point to note is that the liver counterpart of hexokinase, called glucokinase, is not inhibited by glucose 6-phosphate. Glucokinase is instead inhibited by fructose 6-phosphate. This is an example of a feedback inhibition. For both glucokinase and hexokinase, glucose is a stimulator, marking that more must be converted. Coming to the hormonal regulation of hexokinase and glucokinase. The hormone insulin is produced when glucose is abundant in the blood. Thereby, we want to convert more glucose into glucose 6-phosphate. Insulin enhances the transcription of the gene coding for hexokinase or glucokinase. Thereby, more glucokinase or hexokinase is being produced depending on the cell type. If it's a liver cell, more glucokinase is being produced, and in any other cell, insulin stimulates an increased hexokinase production. Glucagon, on the other hand, is a hormone produced when the blood glucose levels are too low, so converting more glucose into energy is not our highest priority, because we first want to stabilize the blood glucose by increasing the glucose molecules in the blood. So we don't want it to undergo glycolysis. Thereby, glucagon inhibits transcription of hexokinase and glucokinase. The second point of control is at phosphofructokinase 1. Phosphofructokinase 1 catalyzes the conversion of fructose 6-phosphate into fructose 1,6-bisphosphate. This reaction is irreversible, and therefore by this point the original glucose molecule has been fully committed. This makes regulation at this point one of the most important and is the reason why it has mechanisms to be both inhibited and enhanced. Let's talk about phosphofructokinase enhancers. When the cell uses ATP, ATP is being converted into ADP. Therefore, the ADP levels rise when the cell is using energy. This makes ADP an ideal allosteric activator of phosphofructokinase 1, since we want to produce more energy. Let's talk about the next regulator of phosphofructokinase 1. Under normal conditions, fructose 6-phosphate should be readily converted into fructose 1,6-bisphosphate. However, when fructose 6-phosphate accumulates, our second enhancer, fructose 2,6-bisphosphate, can act as the most potent allosteric activator of phosphofructokinase 1. But what is fructose 2,6-bisphosphate? Well, when fructose 6-phosphate builds up, some of it is converted into fructose 2,6-bisphosphate by phosphofructokinase 2. This molecule enhances phosphofructokinase 1 activity, promoting the conversion of fructose 6-phosphate into fructose 1,6-bisphosphate and preventing unnecessary accumulation. Later, fructose 2,6-bisphosphate can be converted back into fructose 6-phosphate by fructose 2,6-bisphosphatase, allowing it to re-enter glycolysis. Please also note that both phosphofructokinase 2 and fructose 2,6-bisphosphatase are dimer enzymes. But how does the body know how much fructose 6-phosphate must be converted into fructose 2,6-bisphosphate? Well, the answer is regulation, however this time hormonal regulation. Insulin upregulates phosphofructokinase 2, caused by dephosphorylation, and downregulates fructose 2,6-bisphosphatase, also caused by dephosphorylation. This means that when blood glucose levels are high, insulin is released. This leads to the upregulation of phosphofructokinase 2 and the downregulation of fructose 2,6 bisphosphatase. This will lead to more conversion of fructose 6-phosphate into fructose 1,6 bisphosphate, promoting the glycolysis pathway to progress when blood sugar is high. Contrastingly, when blood glucose levels are low, glucagon is released, which leads to the downregulation of phosphofructokinase 2 by phosphorylation and upregulation of fructose 2,6 bisphosphatase also by phosphorylation. This will lead to much less conversion of fructose 6-phosphate into fructose 1,6 bisphosphate, preventing the glycolysis pathway to progress when blood sugar is low, because our body's priority is on balancing the blood glucose levels. Let us now talk about the inhibitors of phosphofructokinase 1. The first one is ATP, directly contrasting to the enhancer earlier discussed called ADP. The accumulation of ATP means that the cell has plenty of energy and doesn't need to prioritize energy production. Therefore, it only makes sense that ATP is an allosteric inhibitor. The second one is citrate. Citrate is a product of the Krebs cycle, a cycle that comes later in the metabolic pathway, and uses the product from glycolysis. Hence, the accumulation of citrate indicates that energy is being produced in good quantities, and therefore more is not needed. This means that citrate acts as a good allosteric inhibitor. Both inhibitors are forms of feedback inhibition, since they both come from later points in the pathway. Phosphofructokinase 1 is like a metabolic traffic light, controlling the flow of glucose breakdown based on what the cell actually needs. If energy is low, it speeds up glycolysis. If energy is high, it slows it down. The third and final control point lies in pyruvate kinase. The function of pyruvate kinase is to convert phosphoenolpyruvate to pyruvate while making ATP. Similar to the control point at phosphofructokinase 1, this control point can also either be enhanced or inhibited. Let's talk about the inhibitors of pyruvate kinase first. Naturally, when ATP is high, the further progressing of glycolysis is not needed. High levels of ATP will therefore inhibit the activity of pyruvate kinase. Long-chain fatty acids and acetyl-CoA are all products of later pathways that both indicate high levels of energy production. Both act as allosteric inhibitors for pyruvate kinase. Finally, of course, the hormonal regulation coming from glucagon, which is released when blood glucose levels are low. Low blood glucose levels means less glycolysis must happen, since we want to keep the glucose in the blood. Therefore, glucagon causes a reduced activity of pyruvate kinase by causing its phosphorylation. Let's now move on to the enhancers of pyruvate kinase. Fructose 1,6-phosphate is an allosteric enhancer of pyruvate kinase. As you may remember, fructose 1,6-phosphate is a product from an earlier point in glycolysis. When an earlier intermediate from the pathway stimulates a later enzyme to enhance efficiency, it's known as a feed-forward reaction. Insulin acts as an enhancing hormonal regulator, contrasting to the consequences of glucagon. High blood sugar means that more glycolysis must happen. Therefore, insulin causes the increased activity of pyruvate kinase by causing its dephosphorylation. As a little help to remember the enzymes involved in glycolysis regulation, here is the mnemonic we use. Hungry pirates party. Hungry for hexokinase or glucokinase, pirates for PFK1, phosphofructokinase 1, and party for pyruvate kinase. And finally, we've reached the end. Well done for making it this far, as it is a complicated cycle. But if you didn't understand something, don't worry. let's go through the topic in the form of questions called Bobos Brain Busters. Here, we will ask 5 questions. After each question, please pause and try to answer it for yourself. If you need help, go ahead and look back at the relevant parts of the videos. Timestamps are in the description and mentioned in the start of the video. The first question of Bobo's Brain Busters is, how do insulin and glucagon regulate glycolysis through phosphorylation, and how does this affect key glycolytic enzymes? And the answer is, insulin is present when blood glucose levels are high, which activates Phosphofructokinase 2 and Pyruvate kinase by dephosphorylating and promoting glycolysis. And glucagon is present when blood glucose levels are low, which causes phosphorylation of the enzyme, which inhibits Phosphofructokinase 2 and Pyruvate kinase, slowing down glycolysis to preserve glucose. Question 2 is, what is allosteric inhibition and how do ATP and citrate use this mechanism to regulate Phosphofructokinase 1? And the answer is, allosteric inhibition occurs when a molecule binds to a site other than its active site, changing the enzyme's shape and reducing its activity. ATP inhibits Phosphofructokinase 1 when energy levels are high, slowing down glycolysis. Citrate inhibits Phosphofructokinase 1, signaling that the Krebs cycle is well supplied and reducing the need for glycolysis. Question 3 is, what role does fructose 2,6-bisphosphate play in glycolysis, and how is it regulated by Phosphofructokinase 2 and fructose 2,6-bisphosphatase? And the answer is, fructose 2,6-bisphosphate is the most potent activator of Phosphofructokinase 1, ensuring glycolysis proceeds even if ATP levels are moderate. Phosphofructokinase 2 produces fructose 2,6-bisphosphate, enhancing glycolysis, while fructose 2,6-bisphosphatase degrades it, slowing down glycolysis. Insulin increases Phosphofructokinase 2 activity, boosting fructose 2,6-bisphosphate and glycolysis, whereas glucagon increases fructose 2,6-bisphosphatase activity, reducing glycolysis. Question 4 is, how does feed-forward activation ensure glycolysis continues efficiently, and which molecule is responsible for activating Pyruvate kinase? And the answer is, feed-forward activation occurs when an early intermediate activates a later enzyme to ensure a smooth pathway progression. Fructose 1,6-bisphosphate, produced earlier in glycolysis, activates Pyruvate kinase, ensuring the final step of glycolysis proceeds efficiently when glucose breakdown has started. And now coming to the final question, question 5 is, what are the 3 key regulatory enzymes of glycolysis, and how do their inhibitors and activators determine the rate of glucose breakdown? And the answer is, hexokinase is inhibited by glucose 6-phosphate via feedback inhibition. Phosphofructokinase 1 is activated by ADP and fructose 2,6-bisphosphate, but inhibited by ATP and citrate. Pyruvate kinase is activated by fructose 1,6-bisphosphate via feed-forward activation, but inhibited by ATP, acetyl-CoA, and glucagon via phosphorylation. And that's all for glycolysis regulation. We hope you passed your exam with flying colors. If you were also impressed by Barry the civil engineer Beaver, or maybe if Phosphofructokinase 1 is your new favorite enzyme, give us a like. Don't forget to subscribe and ring the bell so you don't miss out on more biology breakdowns. Got questions? Wild metabolism facts? Drop them in the comments. Thanks for watching Bobos Biology. We'll see you in the next one, and stay curious.